Good day. This is the Professor, CBMD PhD. And today's Monday, May 22nd, 2023. It is 9:53 and 34 seconds, p.m. Eastern Daylight Time. And today's research, particulate matter. So let's jump to it here. Fine particulate matter catalyzes oxidative stress in the lungs. Date May 22nd, 2023. Source: Max Planck Institute for Chemistry. As study sheds new light on the adverse health effects of air pollution, hydrogen peroxide production of fine particles may not be as important as previously assumed. A new study reveals that the adverse health effects of fine particulate matter 2.5 are attributable to the conversion of peroxides into more reactive species, such as a hydroxyl radical, OH, rather than the direct chemical production of hydrogen peroxide, H2O2, as previously thought. As based on simulations, they think that the overall concentrations of these reactive species in the lungs are large anyway and not directly dependent on levels of air pollution. They use a computer model to understand the relevant physical, chemical, and biological processes and quantify the health effects of different types of air pollutants. The new model simulates the chemical reactions that happen in the respiratory tract for the first time. They included production, diffusion, and removal of hydrogen peroxide from cells in the bloodstream into the computer model. The scientific investigators say this was quite challenging because it is not so easy to put these processes in biological tissues into equations. The findings of this study suggest that the current paradigms for assessing the differential toxicity of individual particulate matter 2.5 components need to be critically reassessed. The study process of the chemical production of superoxide and H2O2 in a cell-free assay may not be a suitable metric for assessing the differential toxicity of individual particulate matter 2.5 components, and some acellular oxidative potential assays may not capture the actual deleterious effects of particulate matter 2.5. However, the production of hydroxyl radicals, OH, was strongly correlated with Fenton chemistry of particulate matter 2.5 in the model calculations. The model simulation suggests that particulate matter 2.5 mostly acts by conversion of peroxides into highly reactive OH radicals. As thus, particulate matter 2.5 is not so much the fuel, but rather the catalyst of the chemical reactions that cause damage to cells and tissues. Researchers explain the role of inhaled particles in the model. Additionally, particulate matter 2.5 may simulate the production of superoxide from endogenous sources, which further contributes to the adverse health effects of air pollution. The study underscores the importance of continued research to better understand the chemical mechanisms underlying the health effects of air pollution and to develop effective strategies to mitigate these effects. The overall summary: air pollution is a major health risk that affects millions of people worldwide, but the underlying chemical mechanisms are not yet fully understood. Fine particulate matter 2.5 typically contains chemical components that can trigger oxidation reactions. When inhaled and deposited in the human respiratory tract, they can induce and sustain radical reaction cycles that produce reactive oxygen species in the epithelial lining fluid that covers the airways and alveoli in human lungs. Numerous studies have shown that excess concentrations of reactive oxygen species like hydrogen peroxide, H2O2, and hydroxyl radicals, OH, can cause oxidative stress, injuring cells and tissues in the respiratory tract. And this is a Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.